Hey guys, so today we're going to go ahead and start talking about 11.3, exploring basically non-Mendelian genetics. So I'm going to put this up right here. Okay, so genes are more complicated than what Mendel had actually thought. The environment influences the expression of genes, and we call that nature versus nurture. So genes provide the plan for the development, but how the plan actually unfolds depends on their environmental conditions. Some traits have more than just two alleles. So if you're thinking about maybe a pea plant, tall versus short pea plants, we have the dominant T allele and the recessive T allele, and the parent plant just has the two alleles. So they have more than just two allele choices, and that is what we call a multiple allele trait. So an example of that would be our blood types. So we have different allele choices. You have A, you have B, or you have O. So a multiple allele trait, more than just one type or two types of alleles. Some traits are going to be determined by more than one gene. So we call that a polygenic trait. So poly meaning many, gene you see in that many genes. So it's a many gene trait, and it gives us multiple or a variety of different phenotypes. So you just don't have someone that's really short or someone that's really tall. You have a range of people. You have a range of intelligence, skin, and eye color. Traits determined by more than one gene have, like I said, many in-between phenotypes. So there's not just smart people or dumb people. There's a whole range of intelligence between the two. So we have different kinds of dominance. So we've seen complete dominance, and that's going to be where we have completely dominant over another, so dominant recessive relationships. We have incomplete dominance, and you have co-dominance. So we've talked about those in class. So when we have complete dominance, that's what we have typically seen. That's what Mendel saw in his pea plants. The dominant allele will mask the recessive. The pattern, typically the recessive allele returns in a 3-to-1 ratio in the F2 generation. So if you remember, the F2 generation is this generation down here. This is the F1, and we have then the parent generation. Incomplete dominance is a little bit different. We don't have basically you're not completely dominant over another. You see a mix of two traits. So you don't see the expected 3-to-1 ratio in the F2 generation. The heterozygous organisms with the one dominant and one recessive allele show a blend in between the traits. So incomplete dominance, you see basically a mixture, so a blend of the two. So the red and the white flower is going to create pink. Codominance is a little bit different. So both traits are going to be expressed at the same time in the heterozygote. So there's no blending. You actually see both. So this is an example of a roan horse, and if you look at it closely, you're going to see both red and white hair in it. You don't just see like a brown, or not a brown, the red or white. So it's nice, not blend of traits, but you see both of them there. So you see white and red. We also see codominance in our red blood cells. So codominance meaning, so if you look at a red blood cell will actually look over here, or if you remember that all of our cell membranes have glycoproteins on the outside of them. And these glycoproteins help recognize self. Blood types have more than two allele choices, making them a multiple allele trait. So the pattern of sugars that is attached is determined by genes, and we have our different allele choices, like we said before, was A, B, and the O phenotype. It's what's expressed. So in an individual, let's say that this is a red blood cell. If a person has the phenotype in A blood type, they're going to have the A glycoprotein on the surface of their red blood cells. So it's going to look something like this. If they are a B blood type, so just the B blood type, so that phenotype, they're going to have the B glycoprotein, just like this. However, if they're the O blood type, we don't see anything on the outside of their red blood cells, so there's no proteins. So A and B are co-dominant. So if you remember what co-dominant means, both will be expressed. So if both are expressed, what we should expect to see is the A allele. Okay, and we should expect to see the B allele on the out, not alleles, but the types, the glycoproteins, on the outside of the red blood cell. And so that is what we see. So it's codominant. So blood types and the alleles that they make up. So here's our different genotypes. So if you're an AA individual, you're going to have the A phenotype. If you're an AO individual, you're going to have the A phenotype. So if you look there, A is going to mask the O. Okay, so basically A is completely dominant over O. And the same is going to be said for B when it's looked at with O. So we have the BV genotype, it's going to give us the B phenotype. When BO genotype will give us the B again because it's completely dominant over the O. So the O is always going to be recessive. It's going to give us the B phenotype. OO individuals will be an O phenotype, and AB individuals will be an AB phenotype. Okay, so this is an example of not only complete dominance, because we have A and B being completely dominant over A. We also see a multiple allele trait, so you see more than just the AB phenotypes. You see AB, you see O, and you see the AB. We also see complete dominance, or codominance, sorry. So codominance, we see both A and B alleles expressed in the phenotype. So these are just examples of donor bloods, and we're actually going to do a cross and look at people with different types of blood. So let's do a cross between an A individual and an AB individual. Okay guys, so now we're going to go ahead and do a cross between an individual that has the A genotype, phenotype. So the A is expressed, and we would say that they have that A phenotype, with an AB phenotype. Okay, so that's what we would express there. Typically when we're talking about blood, because that we see different relationships in it, we are going to show individuals that have an A phenotype. We use the letter I. I'm not quite sure why we use the letter I, but it helps us see the different types. So if an A individual has an A phenotype, they can be an I like this with the A as a superscript, and they could also, remember we have two alleles for each trait, we could see it like this. Or we could see an A individual expressed like this, a dominant I with an A and then a recessive I. So we can see that we have complete dominance. This recessive I, when we see two of them, would equal an O individual. So recessive I equals an O phenotype. So an AB individual will only have one type of genotype expressed. So we have the dominant I's, and remember they are codominant, meaning that they're both going to express their alleles. So that's why, okay, so we represent them with the dominant I, but then we give them the superscript A and B, showing what they are giving us. So let's say that the two people we're going to cross is this person here. So they have their homozygous for the A allele, and then we cross them with the AB. So what we do is, this is a monohybrid cross, so we're going to go ahead and make our Punnett square. So something like this. And we are going to, through meiosis, we're going to separate their alleles into the gametes. So for this individual, we have the I with the superscript A, I superscript A, and then for this individual, through meiosis, you're going to separate these two. We have I superscript A, I superscript B. And then similar to any Punnett square that we've done, you would just complete the Punnett square. So we have I, A, and here. Okay, so if you remember, and we should, all of these went down, all of these went down in these boxes, and all of these went across, okay? And we have now our different genotypes, our predicted outcomes of the offspring between these two individuals. So now let's go ahead and write their genotypes and their phenotypes. So we have 50% of our individuals will have this phenotype, the 2 dominant I's with the superscript A's. Okay, and then we have 50% having this phenotype, because we see, again, 2 of 4, or 1/2, so it's 50%. So that's our genotype. So then what we have to say is, what's the phenotype? Well, we know if we have 2 A's, that 50% of our people are going to be the A phenotype. And then another 50% will be the AB phenotype. Let's do one more example. And again, guys, I want this into your notes. If you wanted to write anything else in your notes while you watch the PowerPoint, that's perfectly fine, but at least get this in here. All right, so let's now cross someone that is an O individual with, let's say, a AB individual. So we'll look at our Punnett square. So again, we have type, their genotype is the recessive I's with our AB individual, and we have this here. Okay, so we're going to see, again, we see the codominant relationship. So we're going to express the A, B in the phenotype. The O, we're just an O person. There's no glycoproteins on the outside of the red blood cells. So we're recessive, or we put the recessive alleles because the dominant I's will mask the recessive over here. So that's, again, complete dominance when we see one completely dominant over another. So let's separate the gametes through meiosis. So we do that, or not gametes, let's separate the alleles through meiosis. We have the recessive I's, and then we're going to go ahead and separate these two over here. Dominant I, A, dominant I, B. And so then we just complete our cross. A like this, A like this, A like this, B, and this. All right, so let's go ahead and write down our genotypic ratios and our phenotypic ratios. So we have 2 individuals that are dominant I with the superscript A and a recessive I, so that's 50%. Then we also have 2 individuals that look like this. So IB, recessive I, 50%. So now we have to go ahead and take a look at what our phenotypes are going to be. So if you remember, none of these children are going to be O, and the reason why they're not going to be O is going to be recessive compared to the dominant I alleles over here. So it's going to be masked, however, it will still be passed down into the offspring. So the A will mask the O, okay, allele, and so that's why we are going to express this as an A. Oh no, guys, so my video or my computer stopped recording on me. What we did see in that last one is that 50% of the individuals were going to be the, I believe it was A phenotype, and 50% are going to be the AB phenotype. What I would like you to do is the following cross that is posted in this picture right here. Get that done for me in your notes, and that's all you have to do for today. Have a nice night.